Shouldn't be. Headset link. It says it's going. Okay. Is it said it was working. I hate how sometimes these microphones don't. I can back up too. Let me just start at the top. Let me just reback it up. Okay. Again, unit 4, what I was saying, I'm seeing it's working now. Okay. Unit 4 covers chapters 11, 14, 15, and 16. So we're skipping 12 and 13. This is doing protein synthesis as well as then evolution. And I think that these chapters go really well together because we need to understand protein synthesis in order to understand evolution as even the small scale to the large scale. So I am going to run through the major concepts. Remember that you should always be reviewing the objectives themselves, your homework, and those check your understanding questions. But we're going to talk about quickly DNA versus RNA, and then protein synthesis as a whole, so that whole process with transcription and translation, and then the central dogma as well. And then we'll get into some of those main concepts from evolution, including the evolution evidence, Hardy-Weinberg, which I know everyone freaks out about math. It's not that bad, and there's only one question on the test. You can use a calculator, and it's fine. Microevolution, or those mechanisms, and then macroevolution and speciation and cladograms. So that is what is going to happen in today's review. If there's anything else that you have questions on that I don't cover during the review today, just send me an email. I will try my best to answer them. You have your test on Monday, so there's plenty of time over the weekend to study and be like, Ms. Larson, I need help on this one, okay? So DNA and RNA, okay? One of the biggest things we have to keep in mind, and we have to just like cement this in our brains so strongly, is that DNA codes for information. It doesn't do any of the work, okay? RNA does the work. And so I love this little comic from the Amoeba Sisters, because when you think about watching, like, I don't know, Hell's Kitchen, and Gordon Ramsay is in there, he's made the dishes, and then he's just yelling at the sous chefs, the like wannabe chefs. They're the RNA and he is the DNA. So if you can't make proteins get out of the kitchen, RNA, whether it's the tRNA, messenger RNA, so the transfer RNA for tRNA, mRNA for messenger RNA, or rRNA for ribosomes, are going to be the ones that are actually making the proteins. They're going to be the ones that are actually doing the work. The DNA just has those instructions and provides guidance. As such, we do see both of these as nucleic acids being created in the nucleus. So your DNA and your RNAs are all in the nucleus, and then they leave the nucleus to do their jobs. So we don't really talk about RNA, rRNA, and tRNA being made, but the nucleus is making all of those RNAs, and then those RNAs can leave and go out to be doing their jobs in the cytoplasm. DNA is generally double-stranded and RNA is single-stranded, but they are both made of nucleotides. The biggest difference here is that DNA has cytosine, DNA has thymine and RNA has uracil, and then DNA is a deoxyribose sugar and RNA is a ribose sugar. But they're still both in that nucleotide language, and that is where transcription and translation come into play. DNA cannot directly make a protein. It does not have the tools and skills and requirements necessary to do that. RNA is our intermediary who is actually doing that job. That leads me to the central dogma of biology, okay? The central dogma of biology is that genes are expressed as proteins through protein synthesis. So this idea of gene expression, right, from DNA to RNA to proteins, that's your genotype to phenotype that we learned about in genetics. And it makes proteins, and the proteins then do everything else in your cells. The genetic code of living organisms has a couple of features that are really important. The first is that we have redundancy. That means that mutations that can and do occur don't always affect the amino acid sequence because we have a lot of codons that will code for the same amino acid. That's what genetic redundancy is. It's kind of this robustness to ensure that mistakes aren't being made. Genetic code is also conserved. From bacteria through blue whales, which is kind of like my range of things I like to talk about, but from bacteria through blue whales, those codons all still code for the same amino acids. The amino acids are still the same. This means that this genetic code was so important to the evolution of life on this planet that it was retained regardless what organisms became. So it doesn't matter what kind of evolution factors happened, the basis, that genetic code, that fact that a triplet codon codes for a particular amino acid was conserved or is kept the same regardless of what organism we're looking at. Now we can change up the order of those codons and make new proteins, and that's what we see in evolution. But that conservation of that code was so important that it did not happen by chance so many times over to make the organisms we have today. It happened once and then was retained through all of the organisms we have today. So that is something that I just wanted to make sure I touched on and clarified, and it's probably on your test in some way, shape, or form. Hint, nudge, nudge, right? Okay. In order to go from DNA to RNA to proteins, in order to do this idea of gene expression or the central dogma of biology, we have to understand transcription and translation. Now, here's the deal. I am not asking you to fill out a table on this test. There's no table on this test, okay? What I am asking you to do is match something as either transcription or translation. And that something could be as little as identifying the types of RNA, okay? Or it can be as big as identifying the step itself. But whatever it is, we're going to be able to see keywords and we're going to be able to match those. So there is matching on this test for transcription and translation. So let's go through transcription first. Transcription, and I love the Amoeba sisters for saying that the C comes before L because I used to get these confused. And then they said that in the video. I watched it and I was like, duh, that is like the easiest thing ever. But transcription is the first step. And this is where we are taking DNA to RNA and we're still changing, we are changing the nucleotide language, right? We are writing out that code in a way that can be read. So we are taking DNA and we are using a bunch of different things in order to make an mRNA. So the type of RNA involved here is only your messenger RNA for this level of biology, okay? Your messenger RNA is also your product. That is the end goal. I am trying to make the directions in such a way that the cell can use those directions. The DNA is not usable. It's in a language the cell doesn't understand. And so we have to make a messenger RNA, a go-between, okay, a messenger. DNA to RNA occurs in the nucleus. So if I look at matching and if I see anything that says something happens in the nucleus, it has to be transcription. If I see anything that talks about nucleotides, it has to be transcription. So what do I mean by that? Well, if I say the first step is initiation, which it is for both of these, okay? In initiation of transcription, RNA polymerase binds to a gene sequence, which can be called a promoter, okay, and begins to make mRNA. That is my first step. So if I was someone going through and being like, okay, cool, like how do I determine this? Let's unbold the whole thing here. What are my key words here? What am I looking for? RNA polymerase, gene, something to do with DNA. It could be a promoter sequence or it could say gene sequence, but even just seeing the word gene tells me it has to be DNA, which means it has to be in the nucleus, which means it has to be transcription. See where I'm getting at? And then we are making mRNA. That's it. If I see those words, that tells me that I'm talking about transcription. When I move on to elongation in transcription, I am adding nucleotides to a growing mRNA. And again, if I am using anything with nucleotides, or if I am making a growing mRNA, it has to be transcription because I am talking about building the RNA, okay? I'm going to do the same thing for translation, so hopefully you'll be able to see those differences there. For termination, okay, RNA polymerase reaches a terminator sequence of a gene, and mRNA is complete. Okay. Again, you know, I could write this in any number of ways, but if I see anything with RNA polymerase, that is the protein that makes RNA. I am going to be making RNA, which is what transcription does. If I see a terminator sequence of a gene, if I see gene, whoop, what are we doing there? Stop that computer. If I see gene, okay, doesn't matter what the word is talking about. At the end of the day, if I see the word gene, I am talking about RNA being made. I'm talking about the nucleus. If I say mRNA is complete, okay, so I could put on the exam nucleotides added. I don't have to put a whole big long step. You should recognize that nucleotides added is transcription. If I say nucleotides added to a growing mRNA, great, that's still transcription. If I say nucleotides linked by dehydration synthesis in the nucleus, doesn't matter what I say because that keyword there is nucleotides, okay? We don't have nucleotides in translation. In translation, we are doing a different process. In translation, we are reading, let's change it to reading. We are reading the nucleotide language to build a protein using the amino acid language. Because I like to think of these as languages. I like to think about these as like the cells are talking, right? The cells are talking. And so we're talking about what is the cell doing here? What is that job? What is that process? And if I keep in mind that I'm reading the nucleotide language, I am now taking that code in a way that it can be read and I am using it to build something. So if you see something about building a protein, it has to be translation. Translation occurs in the cytoplasm or on the roughy R, but more specifically, it occurs at the ribosome, which is rRNA. Okay. But I could say, if I wanted to be, you know, picky like that, I could say that it occurs in the cytoplasm. It doesn't matter if I don't say at the ribosome because if it's happening in the cytoplasm, it cannot be transcription. It has to be translation. Okay. The RNAs involved here are messenger RNA, tRNA, transfer, and rRNA, which is the ribosome. All three RNAs are involved in this process. We need all three of them in order for this to occur. And my end product is a protein. Okay? So what's my first step? It's still initiation. But what happens here? Okay, this is where things get a little bit different. So what happens here is that mRNA binds at AUG, which is the start codon, to a ribosome, which is rRNA, and tRNA brings methionine to the mRNA. RNA complex forms. So we would say in this case that RNA complex forms, right? That's kind of when I say all three RNAs are working together. But it doesn't matter because if I say mRNA, right, rRNA and tRNA in the same sentence, I have to be talking about transcription. If I say AUG or if I say start codon, it has to be translation because I'm talking about codons, which are those triplet codons, those sequences there. And then MET, I could say amino acid, right? If I say amino acid, I have to be in translation because I am talking about building a protein. If I say RNA complex, I have to be in translation, okay? In elongation of translation, I add amino acids to a growing polypeptide. Again, I might say something like, I add it using dehydration synthesis. That's cool. The thing that's going to tell me what my answer is that it's amino acids and it's a polypeptide. I'm not going to build a polypeptide in the DNA part, so I'm not doing that in transcription. I am only doing that in translation. And then in termination of translation, we would reach a stop codon, and the RNA complex will disassemble. Disassemble. Typing and talking is like typing and writing on the board. It's not the easiest process. The RNA complex will disassemble and the process may restart. Cool. Again, if I see anything with codon or anything with RNA complex, right, that means it's good. If I say protein complete, it has to be translation. So you'll do matching here. Okay, you'll do matching here. That's not the only question you're going to have related to transcription and translation. You'll have bigger, more connection questions based on how protein synthesis plays a role in central dogma and evolution, or why like mutations can be good or bad, or what kind of mutations exist. But a lot of people get stressed out about the transcription and translation. And I want to just clarify that it is matching based on the keys and codes related to those processes. I want you to understand that they're two separate steps, but they go together and it's a big deal there. Okay. Questions on central dogma, DNA, RNA, or protein synthesis?
I have a question about the difference between, okay, so a stop codon is only for translation.
Mhm.
Is that correct? Okay, and then the terminator sequence for transcription is
Yes.
like before it's translated to an amino acid.
Correct.
Okay.
So think about your chromosomes, right? When I said something, I think the X chromosome, I was talking about how there's 2,000 genes on the X chromosome. We're not going to copy all of those genes at once in one big long line. Each gene has its own little section that says like, here's the start of this gene, here's the end of this gene. And then the next section would be a new gene or a new gene. It's all connected together because it's one big chromosome, but that chromosome is made up of little pieces called genes. So the terminator sequence is like a bunch of A's usually at the end of a gene that says like this is the end. And so RNA polymerase goes, oh, I'm reading too many A's. This means I'm done. I'm done with this gene. I don't need to keep going. And it comes off of the DNA. Where the stop codon is just the 3 letters. It's usually UG something. There's 3. I wouldn't ask you those particularly because I can't remember them off the top of my head. But there's 3 stop codons, and they're going to be those 3 sequences of those nucleotides that just is like the period at the end of the sentence, which means the protein's done.
That makes sense. Thank you very much.
Okay. Yeah, absolutely. Absolutely. All right. Let's talk about our evidence of evolution, and I want to change this to anatomical. Okay, so if you remember in chapter 14, I talk about Darwin and his explorations and how he came to these hypotheses and what natural selection means and all of those things. But the big thing with chapter 14 was to talk about the different kinds of evidence we have that evolution is happening. And these are not standalone. These are not one and done. These are not by themselves. We can't simply look at a fossil and say, yeah, this is absolutely a direct evolutionary relationship. Okay, that is never the case. And we used to rely a lot on like anatomical and fossil evidence, but we also are now relying more and more on molecular evidence because it gives us a better picture of like actual relationships. I want to preface that though by saying there have been times when I took a master's level class where we were doing gene analysis stuff, and tell you what, I don't remember a lot from that, but what I do remember is that depending on what you put in, even if it was the same sequence, depending on what parameters you put in, you would get different evolutionary relationships popping out. So while we do rely on molecular evidence a lot more now than we used to, we still need to consider all of these things in order to determine evolutionary relationships. Okay. But all of these have been used for 250 years to determine that evolution is in fact happening. And hopefully you all watched that Harvard Medical video because it looks like I even linked it on there twice because I was like, please watch this, it's so good. that shows that evolution happening so quickly. And what we're looking at there is we're looking at biogeographical evidence, because when you look at the bacteria, you don't go from 0% antibiotics straight to 1,000. It has to build through those different strips of antibiotics, which means that organisms from 0 to 1 are more likely to be related to one another than organisms from 0 to 1,000. Right? Kind of makes sense when you think about it. So biogeographical is just that when we see organisms that are similar in a similar place and time, there's a good chance that they are probably similar for evolutionary purposes. They probably share relationship. Okay. Fossils are incomplete, but are really great because the older we dig, the older those fossils are. We can then determine age, and we've done a lot of carbon dating and a lot of chemistry to determine age of fossils, which is really cool stuff. Embryo development is actually a really big role in determining classification of organisms. So when we start talking about diversity of life in Unit 5, we do talk about how embryos develop and how those embryos can be a sign of evolutionary relationships, because we often start along a pathway that is good because there's certain things you require in an organism, like mammals need to have 2 eyes, 2 ears, 2 nose, whatever, 2 nose, 2 eyes, 2 ears, nose, that sort of thing. And so we see those develop very early and in the same pattern, and then we see deviation after that. So embryological evidence is huge. And then anatomical evidence is actually the one I'm probably going to test you on the most, because anatomical evidence is a really big role in how we, as normal everyday humans, not scientists, as people who are taking a Bio 110 non-majors science class, are going to look at the world around us and determine who is more related to whom. And we look for those physical similarities and differences. And we do this all of the time, even if we don't think about it, right? When we have young kids and we're starting to teach kids to recognize animals, they get in their head that like dogs are furry, so everything with fur is a dog, right? And it doesn't matter if it's a cow or a cat or a horse. They see something furry and they go dog or puppy, because to them, that is the physical similarity that they're seeing. We do this intrinsically. We look at things and we go, oh yeah, these are similar to one another. They must be the same thing. But when we dig into a little deeper, we can actually see that there are going to be traits that are similar or different. And so we will use these exceptionally, we will use these extensively in chapter 17, 18, and 19. So I need you guys to know these anatomical traits, the physical similarities or differences. There's 3 that I told you about in the lecture: vestigial, homologous, and analogous traits. We know that homologous means similar or same because we talked about homologous chromosomes, homologous pairs, and now we're talking about homologous traits. So homologous traits are same ancestor, different function. So same trait or same ancestor, different function, okay? This is like your dog and cat forelimb, right? This is like your gosh, the forelimbs one is the best example of that. But even like jaws, we look at teeth. And if you look at like dog and cat teeth, they're more similar to one another than they are to us. So that would be a homologous trait. But we all have teeth. Teeth is the thing that we see. When you look at fish, we look at the fins or the gills. We look at these physical structures that play important roles in the organism's physical abilities, but we're going to see that those are going to be modified based on what that organism needs it for. Like bats don't have, some bats, like they don't have really good grasping hands because their fingers are so spread out that they fly. Bats don't grasp with their wings, they actually use their back feet. But we have our graspy hands on our front feet. So when we talk about homologous traits, we see a common ancestry and then deviation from there. Analogous traits then are the opposite, where we have different ancestor but the same function. So this would be like your insect wing and the bat wing. I like to think about these like math equations, right? When we talk about analogous traits, we got to the same answer, but we used a different equation. So if I say that 2 + 2 = 4, 1 + 3 also equals 4. Same answer, different equation. So analogous traits are the same sort of thing. We're going to see like We call them wings because they fly, and that's a word that we use. We looked at it, we saw those similarities. We were like, this makes sense. And then we did more digging and went, well, they're not really the same, and we probably should call them different things, but that's more complicated. And so we just determined them to be analogous traits. Vestigial traits, I think everyone generally gets because people really like vestigial traits and thinking about it. These are your evolutionary leftovers. And I think people just like calling them evolutionary leftovers. But things like your human tailbone, goosebumps, wisdom teeth, whale hind limbs, snake nubbins, these are things that were once present and do suggest common ancestry, but then they've been lost over time. It is much easier to lose traits than it is to gain traits. And so we often see these vestigial traits as a homologous structure that has been lost or changed over time. So those are our 5 types of evidence of evolution. And you absolutely need to be able to define and give examples of the anatomical, and then don't brain dump that because we're going to use that extensively in Unit 5, which is why I'm like, please just remember it forever here. I think that's it for evidence of evolution. That's basically chapter 14 in a nutshell. Because chapter 15 talks about natural selection again, and I know I talk about natural selection in chapter 14, but let's move into this idea of Hardy-Weinberg. One of the things that people get a little stuck on, and it can be really complicated, is Hardy-Weinberg is what we call a null hypothesis. We expect it to be violated. We expect it to be not supported. We expect it to be untrue when we test it. So I have some examples of null hypothesis on here where like cats show no preference for food based on shape. That is not true. If you've ever fed a pet or a kid, or even a kid, right? If you don't cut your kid's sandwich the right way, they're not going to eat it, even though it's the same sandwich. You could say, well, kids don't show a preference for sandwich shape. You know that's not true. That's a null hypothesis. Plant growth is not affected by light color. We know that's not true, but we can use that to say, okay, but if I do this, I will see results. So when we talk about Hardy-Weinberg equilibrium, what they're really saying is that we expect evolutions to always be evolving. But in order to determine if that evolution is evolving, we have to determine what rule or what thing we think is happening is being violated. As such, using it as a null hypothesis can be really beneficial. So this is why the assumptions of Hardy-Weinberg equilibrium that states that a population is not evolving if these things are occurring is very easily violated. Because if we think about it, natural selection is always occurring. The end. There are pressures and not everyone is equally related to those pressures, okay? Mutation is occurring. Genes are changing and genetic sequences are changing, okay? Animals migrate and move. Organisms migrate and move. Populations are not always large. We absolutely see small populations, and random mating does not really occur. Individuals choose mates based on preferences through sexual selection, which is a type of natural selection. If all of these are true, then a population is not evolving. But if even one of these is not true, then the population is evolving. And that's what Hardy-Weinberg's equilibrium and experiment and the equation are meant to show. So why do we care about the math? Well, we need to do applied math. Animals are not doing math, okay? Animals are not sitting there and calculating and going, you know, he has like 75% better genes than the guy next door, so I'm going to mate with him because my babies are going to be good. No. But we as humans like to put values to things so that we can say without a shadow of a doubt that something is happening. And it's not just observational. This is where that scientific method and that data collection comes in. So the Hardy-Weinberg equation is just a way for us to determine fitness and contributions to the gene pool in a way to determine if a population is changing over generations, over time. So all we're really doing here is we're looking at a population of frogs, okay? We'll use the Amoeba Sisters video with their, natural selection genetic drift stuff, okay? If I look at a population of frogs, I could see that there might be light-colored frogs and dark-colored frogs in that environment. If I happen to know, because I've done some breeding experiments and I've done the work to figure this out, dark colored frogs are dominant traits and light colored frogs are a recessive trait. Okay, based on what we know about genetics, then we know that there will be a dominant allele and a recessive allele. Okay, I can then count, if it's simple inheritance, Mendelian inheritance, I can count the number of individuals with the dominant allele in a population, right? I can look at a population, let's fix my spelling here, my typing, and I can say, okay, I'm counting 100 individuals, and of those 100 individuals, 80 of them are dark, right? If I count 100 frogs and 80 frogs are dark, that means that my dominant allele, the population of individuals are going to be having at least one allele being dominant, are 80 frogs out of 100. My recessive allele, I would count the number of individuals with the recessive allele. Now, I'm not considering their genotype. Okay, I'm not considering their genotype because I'm literally asking, if I were to look at their first allele in a sequence, right? They have two alleles, they're dihybrid. They are diploid organisms, okay? They have two alleles in the population. I can just say in their first allele space, do they have a dominant or do they have a recessive, okay? If they have a dominant, they'll be dark. It doesn't matter what their second allele is. If their first space is recessive, we know the second one is recessive, but it doesn't matter because they're going to explore and express that recessive trait. So I can count individuals. Now, I don't necessarily want to go out and count 10,000 frogs, but I could count 1,000 frogs, right? If I count 1,000 frogs, I could count easily the number of light colored frogs in 1,000, okay? Or I could count the number of dark colored frogs in 100, whatever it may be. But if I know that I have 1,000 frogs total, and I can count just the light colored frogs, I can then do basic math to determine how many dark colored frogs I have. So in this case, if I said that I had 100 frogs and 20 of them are light colored, right? That is my Q. So this is Q. Q equals the recessive allele. P equals the dominant allele. And in this case, right, P plus Q equals 1, so 80 plus 20 equals 100, or 0.8 plus 0.2 equals 1. So the allele frequencies through phenotype are just P plus Q equals 1. I will give you a value for P or Q. I will say Q is 0.2. You need to then be able to figure out that P is 0.8, but it's just simple math. I hope we get that, okay? We do know, because of our genetics understanding though, that we don't have just a one allele trait, right, inheritance. We actually have dominant individuals can be homozygous dominant or heterozygous, and recessive individuals are homozygous recessive. And so we actually do need to consider genotype possibilities in a population. Okay, when we consider that, we know individuals, if they are heterozygous, they will have one of each allele. And if we do a Punnett square of heterozygous individuals, we end up with a 1 to 2 to 1 ratio for genotypic frequencies. And this makes sense to cross heterozygotes for this because I know that there's 4 possible alleles being, or 4 possible gametes being made. And if I'm crossing heterozygous individuals, that's going to give me the best idea of what my possible genotypes are in a population. This is where they came up with this equation. So when we look at P times P, or P squared, those are my homozygous dominant individuals. Okay. When I look at PQ, those are my heterozygous individuals. And when I look at Q squared, those are my homozygous recessive individuals. And I get the 1 to 2 to 1 ratio here, right? We just usually don't put the ones in because we're looking at a Punnett square. All this means then is I plug and play. So if I know that P is 0.8, that means this is 0.8 times point. So you should put the zeros. By the way, Canvas will mark it wrong if you don't put zeros. Okay, 0.8 times 0.8, which is 0.64. 0.64, which is to do 2, okay, which is my p-squared, okay. 2.8 times 0.2 is 8 times 2 is 16. 16 times 2 is 32. That's my 2 PQ. And then 0.2 times 0.2 is 0.04 or 4%, and that's my Q squared. All that's telling me is that if I were to look and do genetic testing on my population of 10,000 frogs, which I would never ever do in a million years because that's so much time and effort and money, I would anticipate 64% of the population being homozygous dominant, right? 32% being homozygous recessive, and 4% being homozygous recessive, and 32% being heterozygous. If I look at the next generation then, so if I go, okay, cool, but generation 2, in my second generation, I take another 100 frogs, and I actually find 70 dark and 30 light frogs, I'm going to have different values, because if I put in 0.7 here, 0.7 times 0.7 would be 0.49, right? 0.7 times 0.3 is 0.21, and then times 2 is 0.42. And then 0.3 times 0.3 is 0.09. So I actually see from one generation to the next a change in the genotypic frequencies, which suggests that evolution can be happening, and we can track this from generation to generation. Now, if I find a change in the genotypic frequencies, I might go back and see what happened. Maybe there was a fire and some of the frogs died. Maybe we see that there's a change to the environment and light colored frogs are surviving better and darker colored frogs are being seen by predators and eaten. That would be natural selection. Maybe we see, the first example with the fire would be genetic drift. Maybe we see that green frogs are just, they're the hot stuff, and so they're mating more, and that would be sexual selection. So when we look at the Hardy-Weinberg, we're really looking to see, is microevolution happening, and how can we put value to it in a way that we can then go back and figure out what's happening. So it's just applied mathematics, okay? Don't get hung up on it. If you end up not being able to understand Hardy-Weinberg and you can't do it on the test, it's a 4-point question, do the extra credit, move on. But you guys should be able to plug and play and do, 8 times 8, 8 times 2 times 2. I'm really hoping we can remember basic math there, okay? With that, again, these are our mechanisms of evolution. Natural selection is a pressure on a phenotype. Gene flow is the movement of individuals. Mutation is changes, and genetic drift is chance. You'll be doing matching again on this. You'll see some matching on these mechanisms of evolution with scenarios. If you read the scenario and you ask yourself, does this sound like chance? Does it sound like change? Does it sound like pressure? Or does it sound like movement? You should be able to then identify it as drift, mutation, selection, or flow. That's going to be like the easiest way to study for that one. And you can just, you guys saw that, I think, on even the attendance quiz. I'm pretty sure I had something in there on that. So you guys all did good on that. I'm going to tell you right now, this isn't on the test. Okay, so this is not on the test. I took it off. People were getting really stuck on it and really confused on it. And so I've actually taken it off because I'd rather we understand microevolution and those mechanisms there of that evolution, and maybe even understand how Hardy-Weinberg helps us understand and see those mechanisms at play than the different kinds of natural selection. So I'm not even going to go through this one right now because I'm not testing you on it, which means it's not even on the final. But just keep in mind that everything we're talking about overlaps and is in flux. None of this is standalone. None of this is a one and done. None of this is set in stone and never changes again. That's the point of evolution. If we don't have changes, if we don't have mutations, if we don't have movement of individuals, if we don't have anything going on, then evolution isn't occurring. And that is not what we see. We see it happening everywhere, all of the time. Okay? So that's not on the test. I'm not going to go through it. If you want to go through it again, this is the same PowerPoint as last year, so you should be able to go and watch that last online recording from last year, okay? Macroevolution is speciation. You will be asked to do matching on examples of prezygotic and postzygotic barriers. So there is another matching on there where I'm asking you to look at a scenario and tell me which kind of mechanism it is. So make sure you review those. But the big thing here is that hybrids reduce fitness. Hybrids are not good. Hybrids reduce the genes in the gene pool. Hybrids are often infertile or they die. I think several of you on that quiz asked me about like the gene pool, like how do hybrids affect the gene pool? And my example that I brought up is about coyotes and the red wolves. So North Carolina is the last place where red wolves are found. And I actually, let me see, red wolf population 2025. As of August 2025, the estimated wild red wolf population is 28 to 31 individuals. That's it. 28 to 31 individuals. I have classes bigger than that sometimes, okay? If I am a red wolf and there's only 28 to 30 individuals, let's say there's 30 individuals, okay? We're probably not having an equal proportion of males to females. We probably do not have a 1-to-1, so 15 males and 15 females. We might have like 10 males and 20 females, or 20 males and 10 females. But red wolves are they are monogamous. They are a pair of wolves that raise cubs or pups, and so they actually need to be like a 1-to-1 ratio. Now, if you are a red wolf in the wild, okay, your choices are don't mate or mate with a coyote. Generally, if there's a large enough population of red wolves, they absolutely would never mate with coyotes. Red wolves don't like coyotes. They chase them off. They kill them. They are considered like encroaching on territory. They are invaders. So a pair of red wolves will not tolerate coyotes in their territory. But again, you're a male red wolf and there are no females available. The choices are mate with a coyote or don't mate at all. And that driving force behind all of our organisms is to make babies. So the red wolves have been interbreeding with coyotes and hybridizing. But what that means is they're similar enough species that they can make babies, and those babies can reproduce. But those babies are only half red wolves. And again, coyotes are way more prolific than red wolves are. And so that baby grows up and it's a hybrid. So it only has 50% of the genes of a red wolf, but it can't find another purebred red wolf to mate with. So it will mate with another coyote. Now their babies are only 25% red wolf. And then if they mate with a coyote, it's only 12.5%, and then 6.25%, and then so on and so forth, until they're basically just coyotes with like a sprinkling of red wolf flavor. That is going to be bad. That will remove red wolves, functionally remove them from the wild population, because their genes are now so diluted that they don't actually matter or have any sort of influence on the population. Okay, so that's one of the things that we see happening, and hybrids reduce fitness as a general rule. Okay, barriers are there as fail-safe. If one fails, another should stop the hybridization that occurs. We don't usually just see one by itself, and when they do get overtaken, we can see extinction of species as hybrids reduce that fitness. Allopatric and sympatric speciation, let me just throw that on here because I'm going to tell you right now, I like that as a fill in the blank question. Okay. Allo means different land. And so allopatric speciation is speciation that occurs in different land. And sympatric speciation is speciation that occurs in same land. So we'll see that happening a lot on islands. Sympatric speciation will occur, but it's not as common as allopatric speciation. Because you can imagine if you're in different environments, you're not going to be making babies together. Okay. Last little thing here before I talk about my big connection question are cladograms. So cladograms are important because we're going to use these again through Unit 5. These are going to be playing big roles of looking at those traits. And a lot of times we look at the homologous traits that are shared in organisms. When we look at a cladogram, it's like a tree branch. The older part of the branch, the one with the fewest branches coming off of it, is closest to the trunk of the tree. And if you look at a tree right when it's growing out, it grows from the ends. Those limbs grow from the ends. And so a cladogram is like a tree branch in that way. When we look, the older traits are going to be closest to the trunk, and everything after them has that trait. The newer traits, or the most recently evolved traits, are going to be at the ends of the branches where those new organisms are being found. So when we look at this and we say, okay, let's identify the traits that humans have, we have to move backwards from the human line. So the first trait that humans have is a placenta. The next trait is mammary glands. They have 2 pairs of limbs, and they have vertebrae. Okay, so humans have all 4 traits on this cladogram. Now, if I look at what traits humans have with sharks, okay, sharks don't have 2 pairs of limbs, they don't have mammary glands, and they don't have a placenta. I have to go back in time to the most recent nodule of relationship here, which in this case happens to be vertebrae. That's the only thing that these 2 guys share in common. And that is going to be 1 homologous trait. And then the humans have 3 traits that sharks don't even have. Okay. And if we look at the last question here, who is closer, kangaroos and humans or kangaroos and bullfrogs? Well, if we look, okay, and that's actually kind of a silly question. Let's ask it this way. Okay. If we look, we have to say how many traits do they share in common? So kangaroos and humans share 3 traits. They have mammary glands, 2 pairs of limbs, and vertebrae in common. But kangaroos and sharks share 1 trait, just that vertebrae. As such, kangaroos and humans are more closely related to one another than they are to vertebrae. And if we think about it in terms of what we already know about organisms, kangaroos and humans are mammals, right? Bullfrogs are amphibians and sharks are fish. So we know from our general knowledge and understanding that we would expect kangaroos and humans to be more closely related, but the cladogram lets us put it into terms with those scientific, anatomical, molecular, embryological, whatever those traits are that we're looking at, that we can then say, okay, but they share more traits in common, so they have to be more related. Okay? And then the very last thing I want to go through real quick in the last like 7 minutes I have here is, or 10 minutes I have here, okay? How does protein synthesis connect genetics to evolution? When we think about it, that is the big picture question from this entire chapter. How do we go from genotype to phenotype? How do we go from this central dogma where all of these organisms have this DNA, have this genetic code that is used to build proteins, but it leads to this differentiation, these evolutionary traits that we see in all of these different groups? Okay, there is a lot of vocab that could go into answering this question. I will ask you to use a certain number of vocabulary words, I think it's 4 or 5, when trying to answer this question on the test. So there's a lot of different ways to answer this. You can answer this in so many different ways, and I think I answer it differently every time I talk about it. But basically what we're saying, okay, is that protein synthesis is how we go from genotype to phenotype. Natural selection acts on individual phenotypes, as a result, changing the gene pool because those with higher fitness are better adapted to their environment and will reproduce more, leading to changes in the population over time. That's it. Or we can say that natural selection acts on phenotypes, which are a physical expression of a genotype through protein synthesis. Individuals with better fitness through natural selection will leave more genes behind, resulting in evolution over time. There are literally, I have read so many variations on this answer, but what I want us to remember at the end of the day is that natural selection and evolution does not act directly on an individual's genotype. It does not act directly on someone's DNA. It doesn't scan your DNA and go, yikes. So that's kind of bad. No, it's you're now a light colored squirrel living in a dark colored environment. That is a mutation that is going to affect you in a negative way, and you are not going to have fitness. Okay. Remember too that not all mutations are bad. Some mutations just are. And without mutation, we would not have evolution because we wouldn't see changes in that conserved genetic material that leads to those new individuals that we have. And we wouldn't go from bacteria to blue whales. I think that's all I got for today. Is there anything I didn't cover, Elizabeth, that you're like, please go through this for me?
I think I've got it all, but I'm sure I'll email you with a question later when I'm reviewing.
That's fine.
But thank you for doing
100%.
this.
Shoot me a quick email if you've got it. I will always answer if something comes through my way. And yeah, happy studying. I hope you have a good Halloween. Be safe. If that goes for anybody who's watching this later. I told my other classes today too. I'm like, listen, a Halloween on a Friday on a payday weekend, it's going to be sketchy. So if you don't have to go out driving around, it's probably for the best. So just be careful and have a good weekend. Thanks for coming.